In September 2003, a paper published in the American Journal of Human Genetics described something researchers had been looking for in skeletal muscle biology for over a decade. On chromosome 11, in a gene called ACTN3, a single letter change in the DNA sequence, a substitution at position 577, converting an arginine to a premature stop codon, had a measurable, reproducible effect on the composition of human muscle tissue. People who inherited 2 copies of the functional version of this gene produced a protein called alpha-actinin 3, which anchors the actin filaments in fast-twitch muscle fibers. People who inherited 2 copies of the non-functional version produced no alpha-actinin 3 at all. And when the researchers compared the distribution of these genotypes in elite Australian athletes against the general population, they found something that looked at first glance like exactly the kind of clean, singular genetic explanation that sports scientists and journalists had been hoping to find for years. Elite sprint and power athletes showed the functional variant at significantly higher rates. Elite endurance athletes showed the non-functional variant at higher rates. The gene appeared to distinguish, at the population level, between people built for explosive speed and people built for sustained aerobic effort. The paper was careful. Its authors, led by Dr. Catherine North at the Children's Hospital at Westmead in Sydney, noted the association without overclaiming causation, acknowledged the many other factors involved in elite athletic performance, and specifically warned against drawing deterministic conclusions about individual athletes from a single genetic marker. Sports media, unsurprisingly, did not read those caveats. Within a year, ACTN3 had been dubbed the speed gene across a dozen languages. Within 5 years, it was being sold as a consumer genetic test. And within a decade, it had become the centerpiece of a broader argument, appearing on websites and best-selling books, and eventually in the kind of viral video content that now circulates on every major platform, that claimed genetic differences between population groups, differences distributed along racial lines, explained observable patterns in athletic achievement across different sports. The argument was not invented by bad faith actors. It was constructed imperfectly from real data by real people who were genuinely trying to understand something that does appear to be true in a limited, specific, and easily misread way. Understanding why the argument fails requires understanding precisely what the data actually shows, and the distance between those two things is a distance that contains some of the most important and most misrepresented science of the past 25 years. Begin with what ACTN3 actually does and what it does not do. Alpha-actinin 3 is expressed almost exclusively in type 2 muscle fibers, the fast twitch fibers responsible for generating powerful, rapid contractions. These are the fibers that activate during a sprint, a jump, a clean and jerk, a punch thrown at maximum force. They are the fibers that fatigue quickly and that, in endurance activities requiring sustained output over long periods, are less important than the slower, more oxygen-efficient type I fibers that carry the burden of a marathon or a 5,000-meter race. People who carry 2 copies of the non-functional ACTN3 variant, the XX genotype, which produces no alpha-actinin 3 at all, show reduced fast twitch fiber size and diameter, reduced sprint speed, and reduced overall muscle mass and strength at the population level. This is measurable. It is real. It has been replicated across multiple independent studies. Approximately 16 to 18% of the global population carries the XX genotype, and those individuals are, on average, somewhat less explosive than people with the functional variant when other factors are held constant. What the researchers who identified this association also consistently noted in paper after paper is that the effect size is modest, not trivially small, but far from determinative. A person with the XX genotype who trains specifically for power and speed will outperform a person with the functional variant who does not. The gene is a statistical tendency in a population, not a guarantee in an individual. And crucially, this is the point that most popular coverage of ACTN3 has consistently failed to adequately convey. Athletic performance at any level above recreational participation is determined not by one gene, but by hundreds. The current best estimate from genome-wide association studies of athletic performance is that there are more than 200 genetic variants that contribute measurable effects to the capacity for elite sport. Each of them individually explains a small fraction of the variance. The combinations they form with each other and the ways those combinations interact with training environments, developmental history, nutrition, altitude exposure, sleep patterns, coaching quality, and psychological factors are so complex that population genetics cannot currently generate a useful individual prediction of athletic potential from any genomic data set, regardless of how complete it is. This is the technical landscape within which the race and genetics argument about sports was built. And the specific case where it became most consequential and most visible was the publication of John Entine's book, Taboo: Why Black Athletes Dominate Sports and Why We're Afraid to Talk About It, released in 2000, 3 years before the ACTN3 paper and years before sports. Genomics had the data to directly test the claims it was making. Entine's central thesis was that observable patterns in elite sport, Black athletes' dominance of sprint events, West African ancestry, athletes' overrepresentation in American football and basketball, East African athletes' extraordinary success in distance running, reflected a real and substantially genetic basis distributed along lines that he described as racial. The book was genuinely controversial. It received serious reviews, including positive ones from credentialed scientists. It was also criticized extensively and specifically by sociologists, geneticists, and sports scientists who identified both empirical problems with Entine's specific claims and a deeper structural problem with the framework he was using to make them. The deeper structural problem was the same one that runs through every version of this argument that has been made since. It treats patterns observed in competitive sports results, which are filtered through economics, culture, access, training environments, and the specific historical conditions that produce elite athletic populations in different countries, as if they were direct readouts of genetic potential. They are not. And the specific cases that the race and genetics argument most commonly invokes are precisely the cases where the distance between a population's success in a sport and the genetics of that population is most dramatically illustrated by the data. Take Kenyan and Ethiopian dominance of distance running, perhaps the most frequently cited example in this literature. It is real and extraordinary. Since the 1960s, athletes from these two countries have accumulated an astonishing share of the world's top finishing times in events from the 1,500 meters through the marathon. The question of why has been studied directly at the genomic level by researchers, including Yannis Pitsiladis and his group, who have published extensive findings specifically addressing whether genetic factors explain the East African distance running phenomenon. Their conclusion, stated explicitly in peer-reviewed literature and summarized clearly by Pitsiladis himself, is that the African phenomenon does not have a clear genetic basis. Candidate genes, including ACTN3 and ACE, the angiotensin, converting enzyme gene, whose I allele had been associated with endurance performance and altitude tolerance in earlier studies, have been specifically examined in Kenyan and Ethiopian elite runners, and neither has been found at significantly different frequencies compared to non-athlete control populations from the same countries. The XX variant of ACTN3, which is associated with endurance metabolism, is not elevated in elite Kenyan or Ethiopian marathon runners relative to their non-athlete compatriots. The ACE's ID genotype does not distinguish elite East African runners from controls. The genetic variants that the race and athletics argument most prominently features have been tested specifically in the populations most prominently cited as evidence, and they do not show the expected pattern. What does explain the East African dominance to the extent that the research has been able to determine? The honest answer, produced by a meta-analysis published in PLOS 1 in 2022, and consistent with earlier reviews going back decades, is that no single factor explains it, and the combination of factors that together produce the effect has no simple racial interpretation. Elite Kenyan distance runners are not drawn evenly from across Kenya. They are drawn overwhelmingly from a specific ethnic group called the Kalenjin, whose traditional homeland sits in the Rift Valley province at altitudes of roughly 2,000 to 2,500 meters above sea level. Elite Ethiopian distance runners are overwhelmingly from the Oromo ethnic group, whose traditional lands similarly sit at high altitude in the Ethiopian highlands. The altitude factor is physiologically real and well documented. Growing up and training at 2,000 to 2,500 meters stimulates production of additional red blood cells, increases hemoglobin concentration, and produces adaptations in cardiovascular efficiency that translate directly into enhanced endurance performance at lower altitudes during competition. The training center at Aitan in Kenya, widely described as the home of champions, sits at 2,400 meters. Addis Ababa, where Ethiopian national training programs are concentrated, is at roughly the same elevation. Athletes from both countries benefit from decades of altitude-adapted physiology built up through a lifetime of living and training in this specific environment. Beyond altitude, the research has consistently identified several other specific factors. Kenyan and Ethiopian children in these highland communities have historically walked and run substantial distances to school from early childhood. The figure most commonly cited in the literature is between 5 and 20 kilometers daily, producing an early onset endurance training, stimulus that European children at the same age rarely receive. The socioeconomic stakes of running success in these communities are dramatically higher than in wealthier countries. A successful international running career can transform not only an individual's material circumstances, but those of their entire extended family. A motivational structure that produces competitive commitment levels that training environments in high-income countries rarely replicate. Running is not merely a sport in these communities. It is, for many participants, the most realistic path to economic transformation available to a young person. That motivational architecture shapes who pursues running, how intensively they train, and what percentage of potentially talented individuals within these populations actually channel their full capacity into the sport in ways that have no analog in populations where athletics is one option among many rather than the primary viable pathway to a different life. None of this is a simple genetic story. All of it is a story about specific geography, specific altitude, specific culture, specific economics, and specific training environments that exist in particular places and have concentrated athletic talent in those places through mechanisms that have been studied, that are understood, and that have no simple racial interpretation. The Kalenjin are not simply Kenyan, and Kenyans are not simply African. The Luhya people of Western Kenya, who live at lower altitude and have no comparable running culture, do not produce elite distance runners at anything approaching the rate the Kalenjin do. Within the same country, within the same broad population, the African advantage that the racial framing purports to explain disappears almost entirely when you hold constant the specific environmental and cultural factors that the racial framing collapses together. The case for Jamaican dominance of sprint events is similarly instructive and, in some ways, even more directly revealing about what the racial argument gets wrong. Jamaica is a small island with a population of roughly 3 million people. It has produced a disproportionate share of the world's fastest sprinters, most visibly through Usain Bolt, whose performances between 2008 and 2017 produced records that still stand. ACTN3 frequency in the Jamaican population does show a slightly lower rate of the XX genotype, compared to the global average, which is consistent with a marginal, sprint-favoring genetic profile in the population. But what Jamaica also has is a specific cultural and institutional investment in sprint athletics that is without parallel outside of the United States. Sprint is the national sport in a genuine sense, not merely popular, but culturally central in a way that produces extraordinary early identification of talented sprinters, systematic development programs at secondary schools, a competitive domestic scene that provides elite-level competitive experience to junior athletes, and a social status for successful sprinters that drives intense commitment from an early age. The Champs Athletic Competition, Jamaica's national secondary school track and field championship, is an annual event that draws larger live audiences than most of the world's professional track meets, and that produces the competitive crucible in which Jamaican sprint talent is systematically identified and refined. The genetic signal in the population is real but modest. The institutional and cultural infrastructure built around developing that genetic potential into elite performance is exceptional. When researchers examine the ACTN3 data across populations without the racial frame, what they find is not a story about race at all. It is a story about latitude. Across multiple global population datasets, the frequency of the XX genotype, the non-functional variant, correlates with distance from the equator. Populations in higher latitude environments show higher frequencies of the XX genotype, and researchers have proposed that this reflects natural selection for metabolic efficiency in cold climates, where the XX genotype appears to confer superior heat generation in muscle tissue and superior cold resilience. Advantages that would have been significant in the environments that high-latitude populations have occupied for thousands of years. This is a story about climate adaptation. It is not a story about athletic ability. The variant that reduces explosive power in athletic contexts appears to have been selected for in cold environments because it improves thermal efficiency. The fact that it varies across populations is evidence of local adaptation to different environmental pressures, not of racially distributed athletic potential. The consequences of framing athletic performance genetics in racial terms are not merely abstract. Sociologist Harry Edwards, who has spent decades studying the intersection of race and sport, offered one of the most pointed summaries of the problem when he called the argument that black athletes are physically superior to white athletes a racist ideology camouflaged to appeal to the ignorant and unthinking. What he meant specifically was that the argument converts a social and economic reality, the channeling of black athletic talent in the United States, toward a small number OF sports through a specific historical process of limited economic mobility and culturally mediated investment, into a biological claim that naturalizes that channeling, makes it seem inevitable, and in doing so reinforces the very structural conditions that produced the pattern in the first place. If black athletes succeed in basketball and football because they are genetically better at those sports, then the overrepresentation of black athletes in those sports requires no further social explanation, and the underrepresentation of black people in the ownership, coaching, and executive structures of those same sports similarly requires none. The research literature on stereotype threat, the measurable effect of having one's performance shaped by awareness of stereotypes about one's group, adds a specific and well-documented empirical layer to this concern. Studies led by social psychologist Claude Steele and replicated extensively across different domains show that individuals who are aware of stereotypes about their group's performance show different results on performance measures, depending on whether the stereotype is made salient or not. When black athletes are told a physical task measures natural athletic ability, invoking the racial stereotype, they perform differently than when the same task is framed as measuring sports intelligence. The belief that genetics explains athletic performance shapes athletic performance through psychological mechanisms that have been measured and documented in peer-reviewed literature, which means the racial genetic framing of athletic achievement is not merely analytically wrong, but operationally consequential for the athletes it purports to describe. What does the full picture of sports genetics research actually show taken together? It shows that athletic performance is polygenic, shaped by hundreds of genetic variants, each of small individual effect, that interact with each other and with environmental factors in ways that current science cannot resolve into individual predictions. It shows that ACTN3 is real, that its effect is measurable, that it varies across populations in ways consistent with climate adaptation, and that it is nowhere near sufficient to explain the patterns of elite athletic success that the racial frame attributes to it. It shows that the specific cases most commonly cited as evidence for racial athletic hierarchy, East African distance running, Jamaican sprinting, dissolve into stories about specific geography, specific altitude, specific culture, specific economics, and specific institutional infrastructure when examined at the level of the evidence rather than the level of the racial category. And it shows, in the specific case of candidate gene research on Kenyan and Ethiopian runners, that the genes the racial framing most prominently features have been tested in the populations most prominently cited and have failed to produce the expected signal. What we are left with, after 25 years of increasingly precise genomic research into athletic performance, is a picture that is considerably more complex and considerably more interesting than the racial framing allows for. Elite athletic potential is real. It has a genetic component. That genetic component is massively distributed across more genetic variants than anyone can practically measure or interpret. And the conversion of genetic potential into elite performance is inseparable from the specific environment, the altitude, the culture, the economics, the institutional infrastructure in which that potential develops. The patterns we observe in sport are real. The racial explanation for those patterns is not because the question is forbidden, but because the data, examined carefully, does not support it. What the data supports is a story about climate, altitude, culture, economics, and the specific ways that different communities have channeled human physical potential in different directions, for reasons that have everything to do with history and almost nothing to do with biology as a direct causal mechanism. The speed gene is real. It just doesn't explain what people wanted it to explain. And the distance between what people wanted it to explain and what it actually explains is precisely the distance between a story about race and a story about what human beings can do when the conditions are right. Subscribe, because the evidence is always more interesting than the shortcut.